All right, so I'm going to get us started. Before we get started, just so everyone is aware, today's session is being recorded. That recording will be shared with all registered participants. Also, will be available on our New York Sea Grant Seaweed resource webpage, and I'll have the URL for that up in just a minute. For those who are not familiar, my name is Mike Ciaramella. I'm the Seafood Safety and Technology Specialist with New York Sea Grant and Cornell Cooperative Extension, and I'm currently leading a USDA food safety outreach program funded project to develop a hazards and controls guide to support safe seaweed production and marketing. For today's session, I'm going to start off with a brief introduction and update on the project itself before passing the screen to my co-authors who will be presenting on the microbiological hazards chapters of the guide. So the project to date, the purpose of our project, I want to clarify, as we just got back some internal or external review and reader comments, and it was clear that some were a little unclear as to what was the ultimate purpose and intent of this project. So I want to clarify that to start off. So really the goal of our resource that we're developing is to assist industry and agencies in assessing and mitigating risks associated with seaweeds and seaweed products that are destined for human consumption in the US. That includes all seaweeds that are being produced for consumption in the US, regardless of whether they're domestically produced or foreign source product. In all their forms. This ultimately is meant to serve as the foundation for a more specific seaweed food safety training that the team is looking to explore developing once we have this resource in place. So with that, we'll go into specifically where we're at with the guide. So all those in yellow, our first 4 steps highlighted here are complete. We've gone through the chapters, drafting the chapters, did an internal review, and shared those for external review. Received 15 external reviewer comments and feedback representing various agencies, industry, and academic institutions across the US. And the team of core authors and leads is working through the final revisions of each of those chapters as we speak. We're then going to be entering that final stage where we're looking at final copy editing and ultimately formatting of that guide for consistency, also the drafting and development of some artwork for that. So ultimately, those final steps, we're moving towards the final formatting and publishing, which includes the development of some cover art and illustrations to complement some of the contents in that guide. We're going to be getting 2 infographics developed, one that's focusing on iodine, more consumer-centric infographic looking at iodine content in seaweeds, and another one that's focusing more on the importance of addressing some of these food safety hazards as a farmer or a harvester to enable you to better market that product for those who must comply with current prevailing food safety regulations. We're entering then, as soon as we get our reviewers' comments addressed and our final chapters edited, we're going to enter that formal copy editing and formatting phase, ultimately planning for publishing this resource in September of 2025. In addition to that, we have our webinar series. We had our first webinar focused on regulations. That webinar is available on our Seaweed resource webpage. The URL is on this slide here. This is our second, our biological hazards webinar, and then we'll have a third and final in the series focused on chemical hazards in July, on July 11th. All of those will be recorded and available on that same Seaweed resource page. Again, this will be sent out to everybody in an email so you can easily access that information. So that concludes my update on the project as a whole. So now I'll pass the screen to my colleague, Dr. Amanda Shore with Farmingdale State College to kick off the microbiological hazards presentations.
All right, thanks, Mike, for introduction. Again, my name is Amanda Shore. I'm an assistant professor of microbiology, study marine microbiology primarily at Farmingdale State College, and I'll let my 2 other presenters introduce themselves.
Hi, I'm Jen Perry. I am the associate professor of food microbiology at the University of Maine.
And I'm Brian Himelbloom, retired University of Alaska Fairbanks faculty, and I was the associate professor of seafood microbiology. Thank you.
All right, so for this webinar, we're summarizing some of the findings and outlines of the microbial related hazards for this guide. Some of that has to do with the microbial hazards that are maybe present on seaweeds when they're freshly harvested, so introduced at the harvest site, and then others being introduced through potential processing steps. And so we're going to start with talking about foodborne illness, which is really the main focus of these microbial hazards. For the background of thinking about all the food that everyone consumes on a daily basis around the world, that pathogen contamination in that context is pretty rare. But still, we get a lot of foodborne illness cases in the United States and worldwide, up to millions of cases. And we want to reiterate that a lot of these cases or outbreaks go unreported. All right, so people will get gastrointestinal illness, maybe they recover just fine and don't go to their doctor, and thus these cases never get reported in the first place. So if this is the case for a bunch of other types of foods, including land-based agriculture products and seafood products, it's reasonable to assume that this might also be the case for seaweed products as well. So as this industry is scaling and consumption is increasing in the US, it's also possible that foodborne illness could be also on the rise unless we take appropriate preventative measures and controls. All right, so what we do know about outbreaks associated with seaweed products is there have been some outbreaks in the past in different parts of the world. We see mostly norovirus, salmonella, and some more toxic strains of E. coli being the cause of some outbreaks. And we've just outlined these here in tables. So we've got not only cases from Asian countries, we also got here in the US, Hawaii and California, as well as in the UK. All right, and so these were cases that we were able to find easily in the literature. And again, to reiterate that this is probably an underestimate of maybe past seafood or seaweed outbreaks. And that's kind of the impetus for understanding these microbial hazards to prevent future outbreaks. So I want to start off with talking about parasites. All right, so parasites can be a problem with some seafood products, especially fish, some oysters, other types of shellfish. From looking in the literature, we don't see any outbreaks associated with worm or other types of protist parasites specifically associated with seaweeds. So, that doesn't really seem to be a potential hazard that people really need to be worried about right now. I do want to put a caveat out there, though, that two of the more common waterborne parasites that are present in the United States, toxoplasmosis and giardiasis, these are two protist parasites that are present in marine mammals. And so while we don't have any specific outbreaks linked to these two parasites and seaweed products, it's something to maybe consider for the future, since both of these parasites can survive in the seawater for a certain amount of time, and marine mammals can harbor and shed these parasites into the water column and thus potentially contaminate fresh seaweed before it's harvested. So while we don't really have any protocols specifically in federal regulations for testing for these two parasites, it's maybe something that farmers want to consider with their site selection. If we have large populations of marine mammals present, then these two parasites might be something you want to think about. So mostly we're interested in other types of microbial pathogens, so bacteria and viruses. And some of the pathogens that we need to worry about on seaweeds, like in other seafood products, could naturally be marine microbes that are in sea waters just about everywhere. And a lot of these are Vibrio species. Right, so the Vibrios are pretty ubiquitous. There are many different species. The four I have highlighted are some of the more pathogenic species, and all four of these have been detected on freshly harvested seaweeds before. All right, not only are they currently detected sometimes on seaweeds, it's likely that these pathogens may be more problematic in the future with ocean warming, because ocean warming leads to increased populations of Vibrios where they are already established, as well as in some cases allowing certain species of Vibrios to now exist in waters that used to be too cold to support their growth. All right, some of the other bacterial pathogens that we found associated with freshly harvested seaweeds are primarily thought to be contributed to wastewater contamination of the seawater where those seaweeds are grown. All right, so this is a problem we know in shellfish industries where you need to have high water quality and low wastewater sewage contamination in a lot of cases to even put a farm in place. And so some of these pathogens are like E. coli that are producing toxins, Salmonella, norovirus. Those are the ones I have bolded. These things that I don't have bolded are also have been detected on seaweeds, though probably are not going to be as problematic because we don't see as many outbreaks of these in other seafood products. All right, so this is not an exhaustive list of some of the wastewater associated bacteria that could be on seaweeds. And it's certainly not an exhaustive list of the potential microbial hazards from wastewater contamination. These are just the ones that we find most often when I was searching through the literature. All right, so we also wanted to highlight some potential control strategies. And in this case, I'm talking about pathogens that are going to be present in the harvest area. And so really a lot of this is going to be on site selection. So figuring out where to put seaweed lines or cages where we have clean water to prevent some of that contamination from wastewater associated bacteria. All right, so strategies for this. We can just look into the land uses and have some knowledge of the surrounding watersheds, right? If you know if you're near a combined sewage outflow, for example, we may want to reconsider putting a seaweed aquaculture site there. Farms can team up with labs to do direct pathogen detection. You can also work with state and local agencies to utilize their resources for water quality monitoring, right? So some states already have regulations in place where they certify where shellfish can be grown with water quality standards. This could also be expanded to other states in the context of seaweed farming as well. All right, our chapter on this topic has a table of giving some examples of agencies that conduct water quality monitoring and that you can use as a resource. So for example, the EPA, state local health departments, state environmental departments, natural resource departments are some examples of that where you can find this information. All right, I want to take some time to explain that water quality monitoring is done in a lot of different ways. And we can monitor for different types of bacteria when it comes to wastewater contamination. And so these different agencies might use different standards for rating their water quality. A lot of times labs may look for coliforms, which are a rather large group of bacteria that can be indicative of wastewater contamination, but not necessarily. Other places are going to screen for what's called fecal coliforms, which are bacteria that are coliforms, but are more specific in that they are associated with the intestines of warm-blooded animals and are associated with feces and thus wastewater contamination. In a lot of places, there's focusing in more on one particular bacterium, Escherichia coli or E. coli, which is a fecal coliform. And that often can be the standard of water quality monitoring for water samples. But general E. coli detection does not necessarily mean that your product is unsafe because most E. coli is not pathogenic. You really need to start screening for the specific strains of pathogenic E. coli to see if that pathogen is present. And then in some cases, that might be like your indicator of safety. But for a lot of these resources for looking at water quality management for site selection, they may only just look at E. coli or fecal coliforms as a standard. Beyond the coliforms, though, there are other bacteria that indicate wastewater contamination, and so you can use that data as well. This sometimes is linked to beach closures. So Enterococcus is another intestinal residing bacterium that can survive in seawater longer than some of these coliforms. And so sometimes that's an alternative indicator organism for seawater. And state agencies use Enterococcus a lot of the times as their indicator organism for closing beaches and closing other coastal waterways. So if you're don't necessarily have coliform data available, there might be enterococcus data available to help you assess whether your site may have these microbial contaminants. All right, so that was just some of the ideas for these wastewater associated bacteria. For the Vibrios, these are not species of bacteria that are commonly screened for in, say, a state health organization. And so doing specific testing for the pathogenic Vibrios might be helpful to help for site selection. Right, there are many different types of Vibrios out there, so a general screening of Vibrios is probably not going to be that helpful for assessing risk. But screening for the specific Vibrio pathogens may be a good resource to look into. All right, and then for all of these bacteria, the infectious dose is important, where some pathogens, their presence in even very low numbers can be problematic or cause disease. In other pathogens, really high numbers need to be present in order to cause disease. And this is kind of more better established with other food products. This is not an exhaustive list of bacteria that can be problematic at different levels. These are just some examples. And all of these pathogens have been detected on freshly harvested seaweeds. So things like Shiga toxin producing E. coli, that's going to be problematic pretty frequently, even at low levels coming in. Something like Staph aureus, a low level of Staph aureus is probably not problematic. It's really the growth that fuels some of the risk associated with it. All right, I think Jen might be able to speak more on this, but at this point, I will bring it over to her to talk about some of the processes that we talk about in our chapters regarding microbial hazards.
Yeah, thank you, Amanda. One of the things that we really wanted to stress during this presentation is that for a long time, there has been some focus on risk really being equated with whether or not a food product allows the growth of a pathogen. And that is really not correct information for some of the organisms that Amanda just highlighted. For something like Shiga toxin producing E. coli, we can get illness with as few as 10 cells present. So this is a level that we could reasonably expect to see on a naturally contaminated product, and is really one of the reasons that we suggest temperature control whenever possible. So we use this general terminology of the danger zone as established by the FDA, and that is the range between 40 and 140 degrees Fahrenheit, where pathogens will grow most quickly. There are some pathogens, including Listeria monocytogenes, that will still increase in number under refrigeration, but that will happen a lot more slowly. And because of this phenomenon, we really suggest controlling temperature whenever possible, although we do acknowledge that there may be some situations where that's just not feasible. And in those cases, you want to keep product exposure to these temperatures to an absolute minimum in terms of time. Next slide, please. And the reason for this is really sort of what Amanda emphasized at the outset of the presentation. In most cases, we don't expect seaweeds to be harboring these pathogens. And if your seaweed has no Shiga toxigenic E. coli on it, and you don't control the temperature, it still won't have any Shiga toxigenic E. coli on it. But occasionally that does happen. It's something that's been observed multiple times as a result potentially of water contamination, or this is something that can happen incidentally during harvest or transport, even if proper controls are in place. And in those cases, if the temperature isn't controlled, we go from a situation where illness is possible to a situation where illness is extremely likely. And that's really the reason why in any case possible, we want to try and keep the temperature to proper refrigeration, which is 40 degrees Fahrenheit or below in most cases. Okay. Next slide. So the control for this is refrigeration, shockingly enough. We're talking about refrigerated storage or use of any other type of food grade cooling product. Most commonly, we see gel freezer packs because we do want to prevent the contact of seaweed with fresh water that causes quality defects. But these gel packs are really good at keeping the product cold without having any of those negative effects. And so we can either monitor the temperature of our storage environment, keeping in mind, though, that this is not necessarily indicative of the temperature of the product itself, especially depending on the container and the volume that the seaweed is held in. We want to maximize surface area exposure to our cooling medium, whether that's air or whether it is gel packs. Ideally, we are monitoring the product of the temperature of the product itself and trying to keep that at 40 degrees Fahrenheit or below. Or if neither of those are possible, we can actually just look at the cooling packs themselves and make sure that those are staying substantially frozen as a proxy for knowing that the temperature has been controlled. Okay, next slide. Now, moving on to processing, there are a few microbiological hazards that are really very closely associated with particular types of processing or particular types of packaging. And I'm going to touch on two of these. The first is Clostridium botulinum. This is an organism that is relatively ubiquitous in the environment, so it's not unique to seaweed. The reservoir is soil, so anywhere that there can be soil, there can be spores of Clostridium botulinum. But in the spore form, this is not a dangerous pathogen. The issue arises when product is packaged in a way that excludes oxygen. We primarily see this as either vacuum packaging or canning, so in a hermetically sealed package. Under those conditions, the spores can germinate into vegetative cells, which will produce botulinum neurotoxin, the most acutely toxic toxin known to man, which causes botulism at extremely low levels. And so, for example, if you know you're using or there is headspace in the package that hasn't been flushed, this wouldn't really be something that would be a huge concern for you. However, if you are vacuum packaging or you are retorting your product, then you can look at controlling Clostridium botulinum risk with a combination of temperature. And this is the one instance where the temperature recommendation is actually lower. Non-proteolytic Clostridium botulinum, one of the two main subgroups, is actually able to germinate and produce toxin at temperatures as low as 39 degrees Fahrenheit. So if this is part of a control strategy, the temperature has to be monitored and maintained at all times under 38 degrees Fahrenheit or 3.3 degrees Celsius. And this can be combined with a combination of high acidity, so low pH, or high salt levels, which together will prevent the germination of those spores. Next slide. The other really good example of a processing associated hazard is Listeria monocytogenes. And this is an organism that gets a lot of attention in food safety because it causes a very severe illness. The hospitalization rate for people who contract listeriosis from food is upward of 90%. And this is also one of the few pathogens that can actually cross the placenta and cause miscarriage or early term labor. So it is completely unacceptable legally in ready-to-eat foods. That means that by law, any ready-to-eat food has to be free of Listeria monocytogenes in a 25-gram sample. Most commonly, this is addressed through environmental sampling programs, looking at taking samples of surfaces and processing environments through different zones 1 through 4. There is a lot of information about establishing an environmental surveillance program in the FDA's draft guidance on Listeria monocytogenes. But this is something that more or less is considered to be a potential hazard for anything that is passing through a processing environment, just because this pathogen commonly is associated with finding niches in processing environments that are potentially not cleaned and sanitized well. It can establish there and contaminate a pretty broad range of products. It's relatively resistant to acid and to salt, dryness, and heat compared to many of the other pathogens. So we see outbreaks of this organism associated with a really broad range of foods, and it's one of our major concerns. All right, next slide. The control strategies for Listeria monocytogenes in particular, but generally for pathogen contamination during processing and handling, include storage control. One of the main things that we can do is make sure that we're preventing the contact of processed and raw foods. So especially if we are applying a processing control, we want to make sure that that finished product is not coming near raw product, which may be entering a facility with these organisms on it. Applicable only to FSMA, you can also look at sanitation controls, or alternatively, if this is a concern that you have, you can pursue a processing control, which is the subject of the next slide. All right, when we talk about processing controls, a lot of times people kind of use a shorthand for these and call them kill steps. These are otherwise termed lethality processes, and these are processing steps that are used specifically for the reduction of risk associated with a particular pathogen. And the emphasis here is on specificity. So any lethality process that is going to be used as a control has to be validated for a specific product targeting a specific pathogen. You need to be conducting an actual inoculation study using the pack size and equipment that is going to be utilized for applying the treatment itself. So for example, we can't just say you take seaweed and you immerse it in boiling water for this long and now the seaweed is safe, because how long it will take to inactivate those organisms depends on what the organism is. It depends on what the seaweed itself is. It depends on whether it's in a package or not in a package, how large the package is, et cetera. And so all of these things have to be validated in a dedicated study. Usually the processes that are used for lethality are heat-based. So these may be dry heat, steam, or immersion in heated water, but there are other options available, including high pressure processing and irradiation, because these are other processes that are extremely reproducible and can be modeled so that we know that we can expect the same amount of reduction of pathogen every single time that we apply the process. Next slide. This is just a quick summary of what we have outlined today, and I do want to say that all of these topics are covered in significantly more detail in the guide that we are preparing as part of this project. So I would encourage everybody to look through there for more specifics. The important takeaway is really that all of these hazards we've outlined today could potentially be associated with seaweed, depending on what the seaweed is, where and how it's grown, how it's handled, and how it's processed, etc. It's unlikely that all of these hazards will apply to one product, and the key here is to really look at the life cycle of the seaweed itself, taking into account the sanitary quality of the water and which downstream processes are being applied to identify which of these hazards is actually potentially significant for your product. That's it.
Thanks, Jen.
So thank you guys. Yep.
So if anybody has questions, please feel free to enter those into the chat or raise your hand. We'll call on you to unmute. Again, I apologize for our earlier hijacking and anyone who had to sit through or see that. that will be removed from the recording that is shared with everyone. All right. We have one question in the chat from Nicole. How effective is washing in reducing pathogen load in seaweeds?
Anybody anxious to take that?
The answer is we probably don't know. I know from reading some sources like literature that some of these pathogens are still detected even after washing steps. And like, for example, Listeria, some of the Vibrios, even E. coli, didn't completely remove those pathogens after a wash step. So I think there is a lot of work that still needs to be done on that topic.
Yeah, I would agree. I think there's not a lot of research that is focused specifically on the wash step itself, but in most of the studies that have employed a wash step, the numbers in terms of total bacteria really don't go down very much. Things are pretty well adhered to the surface.
Yeah, and I'd add that there's a lot of unknowns still with seaweed or not validated methods, and that what exists out there is a lot of times proprietary, right? There's companies who are doing these things and effectively controlling hazards, but that's not publicly available information that we necessarily have access to and can share, right? So really those more publicly available validated studies through research are what's needed on a lot of these topics to help provide more broad guidance on these hazards. Because I would imagine that what you're washing with can also have an impact, right? You could use salt water or fresh water, and that fresh water we already know can have quality impacts on that seaweed and cause blistering if you're using sugar kelp, right? So there's really a lot that has to be considered when assessing the different strategies for controlling hazards, but also looking at quality. How about?
And I would add to just that the majority of the information in the literature, particularly for domestic product, is focused on sugar kelp. And I think it's really easy for us to fall into the trap of saying, okay, this works this way for seaweeds, but we know that things are very different depending on what species you're actually handling and processing.
Yep. And Jen and Amanda, the next question is really the same, but looking at drying processes. Can drying actually reduce disease organisms?
Drying does reduce the pathogen load on product, but in the studies that have been conducted, and there have been a couple, it doesn't reduce it to the level that we would want to see to say that there's a good amount of promise to use it as a lethality process. I will say, though, that this is largely related to the temperature that the drying is conducted at. And so we almost see this as being more of a heat treatment, honestly, than a dehydration process. But the temperature that's used by different companies is extremely variable. Even with the companies that I've worked with, I would say I'm off the top of my head thinking over a 50-degree Fahrenheit variance in how warm the product is actually getting. But the reduction of water activity itself does not provide sufficient lethality.
Yeah, I think that just speaks back to that. There's a need for validated studies that's more widely applicable. Right now, we don't know for sure, and there's so many different practices, and it really is heat dependent. And I think a lot of the drying practices now are not necessarily high temperature drying that would be considered a lethality step or a kill step, right? It's not intended to kill them, it's just intended to dry those products.
Yeah. I mean, there is product that has validated a drying step at high temperature, but the majority of it is more focused on quality right now. Yeah, and I'll follow up because I see this extra question in here. The studies that I'm talking about have looked both at mechanized drying, so using controlled temperature and humidity dryers, as well as something akin to more of a passive process, so greenhouse drying primarily.
Any other questions? You feel free to raise your hand or put that in the chat. All right. Well, I'll take a minute to reiterate some of what Jen said in terms of the, well, let's take this question first. What do you think about using acid treatment during extraction process of polysaccharides or proteins?
In terms of safety? I honestly would say that with the extracted products, there's kind of a little bit less concern about safety just because of the number of steps that usually are applied to those. So it's not really analogous to like a whole seaweed product.
Did
The
that answer
only I can add that the only studies that I'm aware of that used acid as a control strategy were related to fermented whole seaweed products.
Yeah. Perfect. All right. Another call for questions. For those outbreaks that have been documented, has the contamination been traced back to wastewater?
As far as like tracking it back down to a point source of wastewater, and from what I understand, no. It's simply documenting the physical presence of a potential contamination site, whether it's because that seaweed was grown near an urban area or near a known outfall site.
Yeah, with the most recent, the Salmonella outbreak, that was actually a sanitation issue. Some of the Vibrio outbreaks have been tied into just the presence of Vibrio in the growing waters, which is more and more to be expected, like Amanda said.
We've got time for more questions. If you want to put them in the chat or raise your hand. Are there a lot of seaweed farms, or is this product being harvested from natural beds? Someone who's not familiar with the growing or harvesting of the product. And also, could people grow this in artificial small saltwater tanks? So lots of
Yes,
questions.
all of the above. All of the above. Yeah, it's happening every which way. There are companies that are primarily using wild harvested product. I think the increase that we've been seeing in the past few years is mostly aquaculture operations, but in the marine environment. However, there are also a lot of organizations that are using on land tank cultivation as well.
Yeah, I mean, part of that depends on the seaweed species. Some obviously sugar kelp is a very large species to grow, and it's going to be more difficult to do that in a holding tank, and so the marine aquaculture. But even for the small water tanks, like some of these hazards still from microbial hazards from the harvest area are still probably going to apply because in a lot of cases they're pulling a natural seawater source from a nearby area. I mean, unless you're creating artificial seawater yourself, then some of those hazards may not be the same.
Yeah, and that's
Yeah, and
largely
I would add, sorry,
Go ahead, Jen.
that there is some variability too in the siting for the aquaculture operations. So pretty significant differences in terms of the distance offshore, the depth of the water, and even some of these are happening in estuaries. So even that alone does change the sort of scale of the risks associated with the harvest area.
I was just going to emphasize the global aspect to this. We're talking a lot about domestic production here, but this guide was developed to be a resource for all seaweed product on the market in the US, which is largely imported product that's largely being farmed overseas in various environments. And there's a lot of other species other than the sugar kelp that we're more familiar with our research here in the US. And so the guide was really developed to be a resource for all production and all products that might be making it to U.S. commerce and U.S. market. So it's really important to know what products you're working with when it comes to assessing the risks associated with them and where they're coming from. But yeah, these farming and wild harvest practices are employed domestically, largely with our sugar kelp, but with many species globally.
Yeah, ulva is a great example of a genus that's largely cultured in tanks, particularly in Europe.
Great. Other questions? Well, I'll put a link in the chat to our resource where you can find, or our New York Sea Grant webpage where you can find recording from our regulations webinar. Again, that will be where this reporting is posted once edited. And then you can also register on that website for our July 10th webinar, where we'll focus on some of the chemical hazard chapters. We're really focusing more broadly on biological and chemical for these hazards. But when this guide comes out, there's a total of 16 chapters, so there's lots of hazards covered. Again, making it clear that just because this hazard is in this guide does not mean it is a significant hazard for you and your operation or your state, right? It's really meant to serve and service all seaweed products on US markets, which is largely international and foreign product at this time, but can be a great and valuable resource for our domestic industries to support the development of food safety plans in the current regulatory landscape in supporting safe production of seaweeds, both foreign and domestic. So with that, I'll give you one last call for questions, but if not, thank everybody for joining today. Let me give it a minute for questions. Thanks, Annie.